As our energy problem grows larger, finding new methods of production is becoming a pressing concern. Scientists have begun using hydrogen as an alternative form of energy because of its high energy carrying capacity with virtually no negative effects on the environment. Professor Jim Swartz is currently working on a project that will create a new source of hydrogen using solar energy. I decided to interview Professor Swartz to find out a little more about his project. I am here with Professor Jim Swartz. He's a professor in the Department of Chemical Engineering, and I'm asking him a little bit about his research. Can you give me a brief description of the project you're working on?
So the overall objective is to use sunlight to be able to generate a portable fuel of high quality, and high quality meaning high energy content, but also with relatively little or no environmental impact.
Okay.
And so hydrogen is a very attractive fuel of that type. And so what we would like to do is to capture sunlight and make hydrogen using a microorganism. And to do that, we need an oxygen-tolerant hydrogenase.
Professor Swartz hopes to efficiently convert sunlight and water into hydrogen in a process called photolysis. Solar energy is captured by the photolysis center, which contains photosystems 1 and 2 in an organism. Water is then taken and split into protons, electrons, and oxygen. This process uses a catalyst, a hydrogenase enzyme, which accepts the electrons to continue on and produce hydrogen. However, the oxygen produced in this process poisons the hydrogenase, thereby inactivating it. As Professor Swartz mentioned, this leads us to the bulk of his research, evolving hydrogenase to become more oxygen tolerant. If you could explain to me what the direct conversion concept was?
Right. So that's very important.
Okay.
So much of the whole biofuels excitement right now is taking glucose or some other carbohydrate and converting that into a fuel. But in order to make that glucose, the cell already has taken a fairly large cost. Typically this is a crop cell, a plant, a large loss of efficiency in making that carbohydrate. When we now convert that carbohydrate to the fuel, we take a smaller but still some loss in its energy content in the efficiency of conversion. And so by the time we're all done going from sunlight to the usable fuel, we end up with fairly low efficiency. Right. With the direct concept, we want to use the sunlight to split water and to mobilize electrons, and then to take those electrons directly to the hydrogenase to make the hydrogen. And this promises to be a much more efficient way to go because it's just such a short and direct pathway to the usable fuel.
Right, but the problem with that is that that process creates oxygen and then...
That's right, and the oxygen inactivates the enzyme that we need to make the fuel. So that's why we're focusing on making an oxygen-tolerant enzyme. In order to evolve this hydrogenase, we have to make it into an active form. In order to make it into an active form, we need helper proteins.
Okay.
And so, in fact, we're doing that in cell-free right now. And we're using helper proteins from one organism. We're using the hydrogenase from a different organism. And of course, we're doing all this in an E. coli cell extract, which is yet a third organism. So we're taking pieces of 3 different organisms and combining them and finding that we can still effectively make active hydrogenase.
Where are you going to find these photosynthetic organisms that you hope to use for your project?
There are actually a number that have been studied in microbial laboratories. And there's one in particular, a strain of Synechocystis, another strain of Synechococcus, species that have been studied pretty broadly, and the Synechocystis has even had its genome sequenced.